Now we will transition to the more informal part of the meeting, the management presentation. Now, before I begin, I'd like to introduce two very important members of my team who are presenting with me today. Tom and Lorianne, could you just stand for a minute, just so everyone, you'll be up here in a few minutes. So, Dr. Tom Ulick is our Chief Scientific Officer. He's a board-certified pathologist, was a professor of pathology at the University of California System for 10 years. Following that, he began his industry career at Amgen, where he was responsible for all preclinical development and protein therapeutics, overseeing over 600 scientists and driving all INDs and BLAs during the 1990s, which is arguably one of the most prolific periods in Amgen's company history. After that, he served as founding senior vice president of research and development at Alnylam and helped with the company's successful IPO. He previously worked for me at Conjachem in Montreal as head of R&D. Dr. Lorianne Masioko is a board-certified neurologist and is our current chief medical officer. Before joining our team, she served as senior vice president and chief medical officer at Cubist Pharmaceuticals. There, she managed over 150 employees in areas of clinical research, drug safety, and clinical operations. Before Cubist, she managed numerous clinical trials across a broad number of therapeutic indications at companies that included Nektar Therapeutics, 5 Prime Therapeutics, and Chiron Corporation, which is now part of Novartis. Thank you. So before we begin the presentation, I do remind you that we will be making forward-looking statements. I refer you to all our filings with the SEC to look at the factors that may influence the statements we make regarding our plans for the company. A quick overview. As you know, we are publicly traded, I might add, on NASDAQ. We are dedicated to solely developing effective therapies for spinal cord injury. And we made that decision last July, and I can't begin to tell you how important that has been to provide focus and energy against this one goal for the company. The company was founded 10 years ago, co-founded by Dr. Robert Langer out of MIT, with focus on innovative biomaterials for treating spinal cord injury. We have 2 development programs for acute spinal cord injury. Our neurospinal scaffold, which is in the clinic, is being investigated in the first-in-human clinical trial. For the chronic spinal cord injury market, we are developing a neural stem cell with biomaterial delivery system. We call it our bioengineered neural tissue because there is cell connectivity with that delivery system, and therefore we're literally translating tissue, not just the cells. Now, helping us in our scientific endeavors, we have a world-class advisory team. Bob Langer is still very much working closely with us in our programs. Bob, as you might not know, holds the largest number of medical patents with over 1,000 patents issued. He's the largest holder in the world in that regard. Rich Roberts, who I introduced earlier, is a Nobel laureate in medicine and physiology. Dr. Reggie Edgerton out at UCLA. He was the author last year of the very prominent Brain article, which showed recovery of motor function in paraplegic patients after electrostimulation. Reggie is considered one of the world experts in electrostimulation, which we believe will play an important part in the future of spinal cord injury. And yesterday we announced, we were so pleased that Jim Guest has joined our scientific advisory board. Tom and Lorianne have worked with Jim, and he is a professor of neurosurgery. He's a pioneer in spinal cord cell transplantation for the treatment of paralysis, and we know Jim will continue to add a great deal to our scientific efforts. A quick recap of where we stand. We closed yesterday at $16, bringing the market cap to about $425 million. Primary shares outstanding are 26.6 million. That reflects the reverse split that we implemented earlier this year, and fully diluted, we're at 30.7 million. Our trading volume has always been fairly strong, which helped us certainly in the uplisting to NASDAQ, with average trading volume of approximately 275,000 shares a day. We have cash on hand of $24.5 million, which represents approximately 2 years of cash. To achieve that 24.5, we did our financing, as you know, last May, raising net proceeds of 14.6. We did a raise earlier this year, raising 11 million. And also, due to the increase in valuation, we've had warrant exercises, which have resulted in additional proceeds of 2.6 million. Our monthly cash burn is about 1.1 million, hence my statement regarding the 2 years of cash. I'm very proud of what the team has accomplished in the last 18 months since I've been associated with InVivo. I think you'll see through the introductions and what we've accomplished, we have a superb leadership team with a broad experience base and incredible depth in terms of their knowledge and skills. We've raised cash to fund our clinical trial and the operations. Through our excellent, I think, building of rapport with the FDA, we've accelerated our pilot study timeline. And in that clinical study, we've implanted 3 of the planned 5 patients with our neurospinal scaffold. We announced promising results from the first 2 patients. We've significantly increased our intellectual property portfolio. And one thing that was very important to me, we've increased our publication efforts and certainly the number of presentations we're making in scientific meetings. And last but not least, of course, we did uplist to NASDAQ. You met some of these people today. I just want to acknowledge them all. Lorianne and Tom I introduced. Tomorrow, you know, Stephen McAllister, our Chief Financial Officer. Now, if I could just, the others, just stand up real quick if I want to introduce you, Kristen. So Kristen is our VP of Clinical Ops and Project Management. She's the person who's been out there diligently getting those sites up and running and making our clinical trial possible. Bill? So Bill D'Agostino is our Senior Vice President of Operations. He's responsible for manufacturing and quality management. He's the man to make sure the product is there in the hospitals when a patient presents for implantation of our product. Chris McNulty. Chris heads up our business development efforts, and last year I also tasked Chris with all corporate communications. I hope you can see by the wonderful efforts in press releases and investor relations development, our new website, all that is a great deal that can be owed to Chris. Lisa could stand, please. I talked about our improved rapport with the FDA. Lisa's done a fantastic job in working with the agency. She's responsible for regulatory affairs, and last year I also tasked her with the big challenge of reimbursement planning. It's never too early to start reimbursement planning on a product like ours. There are a few other members of management here. Let me just say, Heather Hamill, just stand up real quick, legal associate. Albert is in our finance department. Peter is in our IT department. Mary Morris is our head of HR. And last but not least, the guy in the back of the room, our Master of Ceremonies, Brian Luque. He works for Chris, working closely with Chris on business development and corporate communications, and certainly does a terrific job in that effort. Have I missed anyone else? I think not. So again, I just think the flavor of the team here is so important in terms of what this group has accomplished. Now, let's move on to spinal cord injury. I think you've all seen this slide many, many times, but just to reiterate, and I think it was Tom that first said it so well, when you think of a spinal cord injury, think of a hammer being just smashed against your hand. It's that type of traumatic injury that is happening to the spinal cord. And on the right side, you see the causes of the spinal cord injury. Now, I think every CEO of every company always says, my product is going to meet a huge unmet clinical need. But I tell you what, I don't think anyone can claim that statement more than InVivo. There are simply no products to treat spinal cord injury. Nothing. There's no drugs. Now, there's some very early work in stem cells that show some preliminary benefit, but they've got a long ways to go yet. But right now, these patients literally have nothing. It's a large patient population, 12,000 new cases each year, which adds to almost 275,000 people living with some form of paralysis. And very few, I think that's what's so tragic, will ever regain any function. The cost is enormous. The cost to treat a patient in the first year alone can often exceed $1 million. And over a lifetime, for example, if a quadriplegic injured at 25, the net present value of that cost is $4.6 million. These are big, big numbers costing our healthcare system, and obviously these are the kind of things we are considering when we talk about pricing, reimbursement, and what the payer strategy is going to be. So simply put, what is our mission? What is the mission of InVivo Therapeutics, and specifically our value proposition for the neurospinal scaffold? 3 major areas we're looking very closely at. First, of course, is the motor recovery, to recover some muscle control, some improvement in movement and strength. Sensory. You know, one thing I think that people often forget is the type of constant pain these patients have day after day, and often have to take narcotic analgesics. So if we can decrease that pain, that's a huge win. And improvement of sensories can help things like avoiding bed sores. They're very hospitalization required. You know, it's just a challenging thing to continue to treat this problem for patients. The autonomic improvement that could be achieved is a major quality of life improvement for these patients. To recover some bowel and bladder control and sexual function truly is transformative for their lives. So at this time, I'm going to bring Tom up, and he's going to walk through some of the early development and preclinical work. And following that, we'll see how that work is translating into the clinic, and Lorianne will review that. Tom?
Hi, everyone. I'm going to talk now about the preclinical and scientific rationale for the use of the neurospinal scaffold in acute spinal cord injury. Here you see a picture of the scaffold. It's 1 centimeter long. You can see it's smaller than a quarter. It's designed in terms of its size to fit into the post-traumatic cavity that forms within the center of the spinal cord after a contusion injury. On the right-hand side, you see a scanning electron microscopy image of the scaffold. You see it's about 85% air, so there's a lot of surface area for cells to regenerate along. The scaffold is made of exactly the same material as bioresorbable suture, which is the PLGA polymer, and this serves as the physical substrate for neural tissue to regenerate along. We have modified that scaffold with the addition of a positively charged amino acid, polylysine, which acts as a glue for the cells and attracts the cells to stick to that physical substrate. The real genius of Bob Langer's design of this scaffold was to promote healing within a cavity or injury in the middle of a solid organ. So we all know that if we fall down and we split open our lip, we go to the emergency room, the physician will oppose the two sides of that two-dimensional wound, either with a suture or something like a butterfly bandage. So when the two sides of the wound are opposed to one another, the cells can grow across the gap and heal the wound. But if you imagine a cavity in the center of the spinal cord, you can't use a suture to sew it together. Number one, because the suture and the needle would destroy the surrounding viable neural tissue, which is very soft. And secondly, when you pull the edges of that three-dimensional cavity together, you would distort and damage the surrounding neural tissue. So Dr. Langer had the concept of putting a three-dimensional bandage, so to speak, in that cavity after the necrotic tissue had been removed, after the dead tissue had been removed. And then the viable living tissue around the circumference of that scaffold would lay down on the physical substrate of the scaffold and slowly grow in with remodeled tissue that could help restore function. And as the tissue grew into the scaffold itself would be bioresorbable, and so it disappears after a month or so. One of the two founding experiments that Dr. Langer did that was the basis for this company was to study the scaffold plus stem cells in a rat hemisection model. And what he found, to his surprise, was that the scaffold itself appeared to have a benefit. After the company was founded, a primate experiment was done it's called the primate hemisection model, in which, as you can see in that illustration, a 1 centimeter long gap of half of the spinal cord tissue was removed and was replaced with the scaffold. When that was done, it was noted, as seen in this image here, this is the normal half of the spinal cord, that within this area of the dashed line, remodeled tissue grew. And the amount of remodeled tissue that grew by appositional healing was greater, significantly so, when the scaffold was placed in the gap. When this tissue, this remodeled tissue, was studied microscopically in this high power view here, you can see nerve fiber regeneration, longitudinal nerve fibers at the points of the arrows, and cross sections of nerve fibers within the circles. The importance of this neural sprouting within the scaffold is that it can create so-called detour circuits that will reconnect. So the sprouting is here in the red, and it can reconnect the existing viable remaining axons to reinnervate muscles below the level of the lesion. When function was tested in this primate model, there was a significant benefit in the gait score of the monkeys as tested on a treadmill using a kinematics methodology. Last year, the company investigated a rat contusion model. The contusion model is a little more representative of the human clinical situation than the hemisection model. What's important to understand in the anatomy of the normal spinal cord shown here is that there's a central butterfly-shaped area of gray matter, and it is surrounded by white matter. The gray matter in the center contains cell bodies and a lot of blood vessels because cell bodies require nourishment and oxygen because of their high metabolic activity. There are much less blood vessels in the peripheral white matter, which are the axons or wires that carry the nerve impulses from your brain down to your muscles in your legs. After a contusion to the spinal cord, the central gray matter becomes hemorrhagic and then is completely necrotic at 24 hours because that's where all the blood vessels are that burst in response to the contusion. But very interestingly, the white matter remains viable for the first 24 hours. So there is a window of opportunity for us to treat patients during this time period by removing the central dead matter, inserting the scaffold, and allowing the existing viable tissue that is important for conductance of nerve impulses from the brain to the lower limbs to be preserved. If one doesn't do anything, the injury progresses via secondary injury mechanisms to cause microcystic degeneration and ultimately cystic cavitation of the remaining white matter. Here you see the surgical procedure performed in a rat. We do a midline myelotomy, cutting into the spinal cord, washing out, rinsing out the dead tissue that you see here. The viable peripheral white matter remains, and the scaffold is implanted in the cavity. Here are two representative sections from a control animal, which was contused, and a scaffold-treated animal. You see large cysts develop in the contused animal at 3 months after the necrotic tissue has been resorbed. This is a fluid-filled cyst. The cyst itself can ascend and cause damages of its own. And the white matter is attenuated by pressure atrophy due to the cyst. On the other hand, when we insert the scaffold is resorbed at 3 months, is replaced by remodeled tissue of the type I showed you in the primate example. And most importantly, there is sparing of white matter that can conduct electrical impulses from the brain to the lower limb. We quantitated this in a large number of animals, and you can see about 80% cyst reduction, increase in white matter thickness, and increase in remodeled tissue that brought the two sides of the wound together. In May of this year, Dr. Jim Guest, who was just introduced as the newest member of our scientific advisory board, gave an oral platform presentation at the American Association of Neurosurgeons in Washington, D.C. and he described the surgical procedure that he developed in the pig contusion model to implant the scaffold. And the procedure that he developed was a precise precedent for the surgical procedure that was used in the clinical trials on our human patients. Additionally, Dr. Guest and his colleague, Nick Theodore, who also performed the pig model and did the first human case, studied the spinal cord tissue pressure with direct insertion of pressure probes into the spinal cord. And what you notice is that at 24 hours after contusion, the normal spinal cord tissue of 10 millimeter rises to pathological levels. And during the implantation procedure, with the egress of the necrotic tissue, the tissue pressure drops and returns to normal at the end of the procedure. This is important because increased tissue pressure leads to ischemia, that is, to compression of blood flow and lack of oxygenation, which leads to secondary tissue damage. So we believe that our novel, really innovative therapy, and we're the only ones in the world doing anything like this, has two main mechanisms of action. One is this scaffold, which serves as a substrate for tissue regrowth and healing, and the second is internal decompression, preventing cell death due to ischemia and lack of oxygenation. So now Lorianne will talk about our clinical program.
Thanks, everybody. I'm very pleased to be able to present today the clinical development program at InVivo. The development program in InVivo is really characterized by speed and efficiency. And so what do I mean by that? The device has received humanitarian use device, which enables us to go very rapidly towards commercialization. And the main characteristics of that is that it's used to treat patients with very serious disabling diseases, but that's also a relatively small population, and in this case, less than 4,000. And we're required to show safety, but also probable benefit. So this is what really distinguishes it from the typical development path. And I would point out that the key phrase in the regulations is that it's probable benefit to health from the use of the device outweighs the risk of injury or illness from its use. And so probable benefit is a much lower bar to approval. So this is a very efficient means by which we can move towards commercialization. So the neurospinal scaffold pilot study is currently underway and doing very well. We've already enrolled 3 of the 5 subjects, and the 3rd subject is currently in follow-up as well as the others. Our primary objective is to demonstrate safety and feasibility of the scaffold, and the secondary objective is to show some preliminary evidence of effectiveness. So what do I mean by effectiveness? Really, we're looking at a number of neurological outcomes that are important to the patient, obviously. But in addition to that, we're also looking at some quality of life measures, things that enable patients to be mobile, to make transfers, bowel and bladder function, which is as important to patients as neurologic function. We're also looking at MRI measures to determine whether or not the scaffold has any appreciable effect on things like cyst formation. Now, the safety and effectiveness is being looked at very, very frequently in the beginning of the study. They're being looked at 24, 48, and 72 hours initially, then weekly, monthly, and then at 12 months. And so I just want to point out that the majority of gains made by patients neurologically, if there are to be any, occur within the first couple of months. And we're looking beyond that because our hope is that the scaffold will continue to provide some degree of efficacy even beyond the first couple of months. We currently have quite a large number of clinical sites up and running, thanks to Kristen Neff and her team. We have 11 centers, and these are some of the top centers in the United States that are very, very busy. They see a lot of spinal cord injuries and traumas to the spinal cord. And these 11 sites are currently looking for our next 2 remaining subjects that we will be enrolling into the trial. So this gives us a lot of optimism that we will actually be completely enrolled by the end of the year. So in terms of what's been happening on the regulatory front since the last time this meeting was held, the last shareholders meeting, there's been a great deal of acceleration in the regulatory front. So what do I mean by that? The FDA had originally required a 3-month waiting period, a holding period after each individual subject. So what that meant was we would be in a holding pattern for at least a year. Forget about the time it takes to enroll patients, but we'd be holding for a year. And in December of 2014, there was a great deal of progress made on the regulatory front in that after the first patient, there would only be a 2-month waiting period instead of 3. After the second patient, there'd only be a 1-month period of waiting, and subjects 3 through 5 could be enrolled concurrently. So that effectively shaved 9 months off of the study timeline. So that's very significant. And again, as I mentioned earlier, we anticipate full enrollment of the 5 subjects by the end of the year. So how have the patients been doing? As mentioned earlier, we enrolled 1 patient at the end of last year, and this patient has been doing extremely well. This patient had a complete spinal cord injury at the T11, T12 level. So what that means was at the bottom of his trunk, he had an injury that didn't allow for any movement, sensory, or autonomic function below the level of that injury. And by the 1-month assessment, the patient had already regained a certain degree of voluntary motor control. And by the 3-month time point, he had really increased his level of function substantially. So he was able to move at the hip joint and the knee joint, whereas previously he had no lower extremity movement. He had a gain of sensory function down into his legs. And I think very importantly that we sometimes forget about, and it's really important, is he had nearly a complete recovery of his bowel and bladder function. So clearly he had made substantial gains. And what's very unusual is that less than 5% of patients with complete spinal cord injury will make any degree of recovery to what's called an ASIA C or an ASIA D in terms of motor recovery. So this person really beat the odds quite substantially. And in addition to that, he continues to improve at 6 months, and that's very unusual. As I said earlier, most improvement occurs in the first couple of months. So the physician who did the surgery, Dr. Nick Theodore, who's been referred to by previous speakers, he's at the Barrow Neurological Institute. He's very pleased with what he's seen with this first patient. The second patient was enrolled earlier this year, and this patient was again found to have a complete spinal cord injury, meaning no neurologic function below the level of injury. At the 3-month examination, although this patient remained an ASIA A, so she remained with a complete spinal cord injury, she also at the 3-month exam had some very important improvements in quality of life, her ability to maintain trunk stability. So all of these things are important in terms of allowing the patient to do transfers from a wheelchair to a car. The patient had significant improvement in bowel and bladder control, and so these are obviously all very important to the patient's quality of life. We very recently enrolled a 3rd patient, and prior to surgery again, this patient was assessed to have a complete spinal cord injury. We're still waiting for the first assessments to come in from this patient. This patient was only enrolled a couple of weeks ago. Both of these patients were enrolled by Dr. Dom Korek at the Carolinas Medical Center, who now has the attribute of having the world's greatest expertise in the neurospinal scaffold. So we're not just waiting for the pilot study to complete. We're also looking forward to developing what we are calling the pivotal probable benefit study. The reason why we call it that is this is not a standard pivotal study where you enroll hundreds of patients and you have a randomized control and that sort of thing that takes years and years to complete. Again, I just want to remind everybody that this is a probable benefit study. So we just have to demonstrate safety and probable benefit. And what we're looking at are important neurological outcome measures such as motor recovery, but we're also looking at those things that the patients consider to be some of the most important aspects of their life. Mark mentioned some of these earlier. We're looking at bowel and bladder function improvement. Again, the patients have significant pain. That's one of the most significant aspects of this condition. So we're looking at whether or not the scaffold can significantly reduce pain, and we're looking at quality of life improvement, meaning everyday living outcomes such as ability to be mobile. And it's important to point out that virtually every one of these outcome measures was achieved in terms of improvement by the first subject. So that gives us quite a bit of hope in terms of the future of the neurospinal scaffold. So again, the pivotal probable benefit study plan, we are going after an HDE approval. We believe that we can use historical controls. There's a lot of precedent for that for HDE approvals. And what that effectively does is it reduces the number of patients that we need to enroll by at least a half, possibly even more. Although the final endpoints will be informed by the pilot study, we do have already a pretty good idea of what this study is going to look like. And assuming continued positive results, we plan to take this in front of the FDA and get their final approval as well. So we're looking even beyond the probable benefit approval for thoracic patients with acute complete spinal cord injuries. We're looking at additional looking at additional populations. So one of the populations that we're actively considering are the acute cervical spinal cord injuries. So these are the patients for whom there is really the greatest degree of unmet medical need. These patients are quadriplegic. And as I mentioned earlier, we have HUD designation for the cervical patients as well. So we can, if we choose to, we can pursue the same very rapid, streamlined HDE approval for this population as well. We're also looking at full approval opportunities via the pre-market approval path, the PMA path. So this is a more traditional ability to expand to the entire acute SCI population. So it's a little bit longer, but it also gives us a greater opportunity to treat a broader population of patients. So that would include the thoracic complete spinal cord injury patients, cervical complete spinal cord injuries, but also a very important group of patients with both cervical and thoracic spinal cord injuries for whom they have incomplete spinal cord injury. So what does that mean? That means that they still have some residual function below the level of injury, but they're still significantly impaired. And this is a population for whom improvements in motor and sensory recovery, autonomic recovery would also be very important. The PMA pathway also gives us the possibility of following an expedited access pathway. And what that means is you get an accelerated type of approval, and some of the clinical development, or a great deal of the clinical development, can be done in a post-commercialization setting. So it allows us to get to commercialization more quickly. You do still need to do some clinical development work, but it's in a post-commercialization setting, which could be very beneficial to us. We're also looking at the possibility of taking our clinical programs offshore, and the clinical and regulatory strategies for ex-US development is currently under active review. And we will also explore the possibility of corporate partnerships for ex-US development and commercialization. So I'll turn this back now to
Thanks to Tom and Lorianne. I think you did a fantastic job giving some really good insight as far as the science and what we're trying to achieve here. I want to move on now to the chronic and just introduce this. I'm going to bring Tom up real quick to talk about the, again, the bioengineered neural tissue. So as I mentioned in my introductory comments, 273,000 people living with spinal cord injury in the United States. Again, very few therapeutic options. Now, the neural stem cell transplantation does offer therapeutic promise. There's some very early data that was just presented in Montreal a few months ago, and that company is now moving into a larger clinical study. And again, this would be in the chronic population. One thing that's been a tremendous change, I think, for this area is that GMP neural stem cells are now available, which totally changes how we look at the development program, that InVivo ourselves don't have to go through a massive scientific program to develop our own neural stem cells. So Tom, why don't you come back up? Why don't you, we'll review just some of our preliminary thinking on how we plan to develop this, the scaffold plus cells, and some other thoughts that Tom and his team are working on.
So, as Mark said, our approach to the chronic spinal cord patient is with stem cells, but we have a very unique and differentiated approach from anybody else, really. We made a couple of strategic decisions about how to proceed with this program. The first is that we would use true neural stem cells that can differentiate into the three lineages neurons, oligodendroglial sites, and astrocytes that are necessary to reconstitute fully healthy neural tissue. There are probably dozens of so-called stem cell trials in the world today, but most of them use either bone marrow-derived stem cells or mesenchymal stem cells or restricted lineage cells or genetically transfected cells. There are very few that use true neural stem cells. The second guiding principle we have is that we will introduce the true neural stem cells directly into the spinal cord at the site of the lesion. This is very different than many of the spinal cord injury trials ongoing right now, which use either an intravenous delivery or an intrathecal delivery of the stem cells. The third thing, which is unique, is that we will use a scaffold to introduce the neural stem cells into the injury site in the spinal cord. We believe that a scaffold, which is a physical substrate for the neural stem cells, is important for attachment, sprouting, and elongation of the nerve cells. This is very similar to the analogy of a flying seed from a plant that needs to attach to the soil, then sprout, and then needs something like a trellis if it's a vine to grow up. So a stem cell is round, but after it attaches to a physical substrate, it sprouts and then elongates axons. And this is illustrated in the next slide with rat neural stem cells. And you can see that right after they attach to a scaffold, they start making little protrusions and elongations, which are called neurites, which will then differentiate into axons and dendrites. And here on a scaffold chip, you see a little later that the many neural stem cells have elongated processes, which are axons, and that the cells have attached to each other with dendrites. Here you see now human neural stem cells that we've attached to our scaffold that we're using for the acute spinal cord injury patients. You can see it longitudinally and in cross-section. And you can see that in the open spaces, a truly artificial neural tissue has been recapitulated. And you see stained in red are astrocytes, and following those structural astrocytic cells are the green stained neurons. One approach we're exploring in our preclinical program is to surgically remove the glial scar tissue at the site of injury because it prevents regrowth of neurons, because it's scar tissue and it's a hindrance to regeneration of neural tissue. And then implanting surgically a scaffold such as the one I just showed you, where the glial scar tissue has been removed. Another approach we have with our bioengineered tissue is to develop injectable scaffolds, which is a more minimally invasive approach. And here's an example of a jello-like consistency scaffold with neural stem cells in it. And you can see how the neurites sprout and grow out after a single day already into a growth matrix. The concept of the neural stem cells on a scaffold is the same whether it's surgically implanted or injectable. And that is, in this cartoon sequence, you see a normal axon innervating a muscle. After injury, the axon is disrupted. We go to the injury site, implant the scaffold with neural stem cells on it. The scaffold is biodegradable and is replaced by nerve tissues that form a detour circuit to reconnect to the muscle and innervate the limbs. This concept was again first illustrated in vivo in an animal model by Dr. Langer's lab. And you can see here, here's the scaffold alone, the neural stem cells alone, the lesion alone, and the best result is achieved with a combination of neural stem cells and scaffold, which is why we're taking that approach. What we do in our laboratories is to study different scaffold biomaterials together with neural stem cells, which we attach in different ways and differentiate in different ways on the scaffold. And then we study that combination in animal models. It's very important to us in this unique approach we're taking to go into the clinic with the optimal product. So we're taking our time to make sure we have that before we file the IND.
Thanks, Tom. Now let's move on to the commercialization, and I want to cover intellectual property and just then to wrap up. Regarding the commercial opportunity, I mentioned earlier the cost to treat these patients and how extraordinarily high it is to our healthcare system and the impact it has. We know that when we aim to look at commercialization plans, that the majority of acute spinal cord injuries are treated at the approximately 200 level 1 centers in the United States. This makes, I think, this product very well suited for us to completely vertically integrate and commercialize the product ourselves if we so choose to. When you look at what is the prevalence and what is the market size of that HUD destination, which will be going after HDE, that would be the acute, complete cervical and thoracic. There's about 4,000, as Lorianne mentioned earlier. That drives a market size of probably about $500 million a year. If you look at all acute spinal cord injury, where we go after both the complete and the incomplete, that's approximately a $600 million to $1.8 billion a year opportunity. And of course, the huge market opportunity is the chronic patient population, those 270,000 patients. If the work that Tom had just described progresses, that patient population will drive a market opportunity well in excess of $10 billion. Intellectual property. I can't state how important this was to me when I first joined the company, that our portfolio is as strong as it possibly can be. And last year, it was just tremendous that Bob Langer himself went down to Washington to the Patent and Trademark Office and helped secure this fantastic patent. It's a broad U.S. composition and methods patent covering both the neurospinal scaffold and the scaffold plus stem cells. And again, this all comes out of Bob's lab, where he, Dr. Rajiv Saigal, and Dr. Tang were the co-inventors of the scaffold. Now, there are a number of additional intellectual property activities as we look for further patents in the work we're doing, things from a range of things from packaging, instrumentation, some of the work that Bill is doing in the manufacturing area, and of course what you just saw that Tom is pursuing. There could be many opportunities for patenting some of the work regarding the bioengineered neural tissue. I mentioned also that it was very important for me to begin to really ramp up publications and presentations at major scientific meetings. This is a way in which you get our word out to key opinion leaders and develop the experience and knowledge base necessary for successful commercialization. As Tom mentioned, Jim Guest did present at the ANS, just a place in Washington a short time ago, on the work that he did in the porcine model. And Dr. Alexander Roper, who with Dr. Nick Theodore did the very first implantation, he presented that work also at the ANS. We have 3 additional upcoming presentations. We'll be presenting at the New England Neurological Society meeting here shortly. In the fall, we'll have the annual symposium of the National Neurotrauma Center, and one of the other very big meetings, the annual meeting of the Congress of Neurosurgical Surgeons. InVivo will be presenting our scientific and clinical data at all these meetings. In addition, we have a number of publications in process. So to summarize, it's been a very gratifying period here. We have our first trial has been initiated. The first patient, as you know, was implanted last October and reported the 6-month data in May. The second patient was implanted in mid-January and reported that 3-month data in May. And as Lorianne mentioned, of course, we just enrolled our 3rd patient. We talked about the acceleration of the pilot trial timeline. I can't emphasize how important a win for that for the company that was. We've got the team now in place to certainly drive these valuable assets. We've raised a considerable amount of money, and we now are sitting on 2 years of cash. We received that excellent patent covering both the scaffold and the scaffold plus cells, and again, the increased publications and presentations. So what are the expected milestones or catalysts for you as stockholders? Well, clearly the 5 patients, each in themselves, represent a potential catalyst. As we've guided all along, we will provide updates on those patients as appropriate, pending whether there's safety concerns or certainly a significant improvement in terms of their effectiveness of the product in their condition. We also, as Lorianne mentioned, want to meet with the agency very shortly here and begin discussing with them the protocol for the pivotal probable benefit study and hope to get that started early next year. I want to thank you again for your attendance and certainly for your support of InVivo Therapeutics.